Welcome, everyone, to the MAPS Canada Research Room. I'd like to begin by acknowledging the traditional owners of the land on which I meet today. I'm mindful that many of you are joining from different territories, and I invite you to reflect on the lands you are on as we begin our meeting. All right. The research room is produced by MAPS Canada, which is a volunteer-run nonprofit charitable organization committed to expanding safe, legal, and equitable access to psychedelics through research, education, and advocacy. The MAPS Canada Research Room is a monthly online program that highlights cutting-edge Canadian research and researchers, or research happening in Canada, from a range of disciplines within the growing field of psychedelic studies. The research room offers a platform for researchers to actively engage with the audience about their current work. We are joined today for a great session with Hailey Gilmore. And Hailey is a master's level biostatistician with over 16 years of experience in clinical research oversight. She has contributed to the life cycle of more than 60 clinical trials and is co-author of 30+ publications and talks spanning several therapeutic areas. The nexus of health, justice, and impact is the ethos for the work she chooses. After spending 11 years managing NIH-funded HIV clinical trials in the US, South America, and Africa, including multiple regulatory approvals of drugs and devices, Hailey came to psychedelics. Since 2020, Hailey has worked at the intersection of ClinOps and MedAffairs for MAPS, PBC, Lycos Therapeutics, and as a private consultant, and for AtaiBeckley, managing programs in the US, Canada, South America, Europe, and Australia. She was an SSDP pipeline mentor for 3 years, and her work has been featured on Psychedelic Alpha and the Trip Report podcast. Welcome, Hailey, to the research room and to all our guests as well. I'll turn it over to Hailey now.
All right, thanks Riel for having me, and thanks to everyone for joining. Looks like it's going to be pretty intimate, so hopefully we can have plenty of time for questions. As always, it'll take me a second to get this up and running. All right, that's all set. All right, you're looking at the wrong screen. I can already tell. Hold on. This is the worst part about presenting, is getting the sequencing right on your clicks. Okay, we're starting over. Well, I guess while I'm here, I will just say that, again, the talk is meant to be just an overview of our pipeline to date at AtaiBeckley. It's not going to be any new information. This is information that likely you've already seen published if you've been following our work. So that's just a bit of a disclaimer that these are all encore data. I will say that, we are hoping to engage with current stakeholders as well as new stakeholders as we proceed into some later stages of our current trials. And so we could use today as an opportunity to do that. So that, again, there will be time for questions and answers at the end for sure. And I think I finally got this right. All right. Yeah, I did. Here we go. So again, I'm talking today about the 3 assets we have in our pipeline. VLS-01 is the program that I was hired to work on. We also have another candidate that's just wrapping up its phase 2 program, EMP, and I'm sure most of you are familiar with BPL-003, which was an asset that we had previously partially sponsored and now have wholly acquired now that Atai has merged with Beckley as of November last year. Standard disclaimer slide. This is a presentation on research, so any statements here are meant to be to present data or not making any claims. As I noted, we have 3 assets currently in development. Our pipeline really is focused on some of the most urgent mental health care needs. We have BPL, as I have stated, is moving towards the phase 3 program. VLS is currently in phase 2 and recruiting, and then EMP-01 has just wrapped up phase 2, so we are going to be pushing out those results fairly soon. I don't have to tell this group that there is an urgent mental need for novel healthcare treatments. There's over 300 million people suffering worldwide from depression. It's the second leading cause of disability in the world, causing a huge economic burden both in the US and globally. What we do know, obviously, is that the treatments have a slow onset of effect, which doesn't work for a lot of people. They want to feel better quicker. And some people who are treated do respond to treatment with our current treatments of SSRIs and SNRIs, and we have some emerging, obviously, innovative psychiatric treatments like TMS or ketamine. But some people still remain with symptoms. A vast majority of people remain with symptoms and may experience side effects from the current treatments that they're on. And we also still have a huge unresolved problem of TRD patients with suicidal ideation. And that's where we're hoping to intervene. So first I'm going to talk about BPL-003 because it is the most advanced of all of our assets. It is a dry powder intranasal formulation of mebufotenin, which you would probably know as 5-MeO-DMT, which primarily acts as a serotonin receptor agonist. It uses a device to deliver the dry powder in the intranasal pathway that is very similar to how Spravato is delivered, or S-ketamine. We were granted breakthrough therapy designation for this product after our positive phase 2b results, which demonstrated a rapid and sustained antidepressant effect after a single dose. There have been multiple phase 1 and phase 2 trials of BPL-003 to date, which led us to feel comfortable about the safety and efficacy profile as we approach phase 3. We did just recently have our end of phase 2 meeting with the FDA. We're taking that feedback into consideration and hoping to launch the trial in the second half of this year. Second quarter, second half. And what we're hoping with this product, as well as a few of the other products in our pipeline, is that we can really hone in on existing clinical care structures that have been built out through the ketamine clinic models. Because we have this shorter acting drug, about 2 hours, that includes, during and post-dose observation, so that any of the infrastructure that's already been built out would potentially easily absorb such a new product. So I'm going to go over the phase 2 results so far in TRD. BPL-003, the most recent 2B design, was randomized, controlled, masked. It was with patients with moderate to severe TRD. There was an open label extension. So one thing to note, like most trials, people had to be willing and able to discontinue their current antidepressant treatment regimens in order to be in the trial. That's a pretty consistent theme throughout psychedelic trials. There were 3 arms in this trial. There was a high dose, a medium dose, and then a low dose control. And in addition to looking at the primary endpoint, which was the drop in or change in MADRS score at 29 days, we were also looking at responder and remission rates and changes in the MADRS over time to see if the effect could be sustained. So the core study results were that it met its primary endpoint of a statistically significant change in MADRS from baseline to day 29, both in the 8 milligram arm and the 12 milligram arm as compared to the active low dose control. You'll see here that the drop was about 12 in the 8 milligram and about 11 in the 12 milligram. So very similar change, so a very similar change or very similar delta from the active control, which led us to think that while there was comparable efficacy between the 2 arms, we may be able to get away with a lower dose and therefore expose people to less drug. And so you'll see what we decided to do with that information as we get later on into the BPL program. Like I said, we also looked at responder and remission rates. So there was pretty immediate and lasting clinical response in more than 30% of our patients, and about a quarter of them still were at remission at about week 8. I'm going to come back to this idea later about looking at high rates of response and remission when we look at the results from the open label extension. For balanced coverage, I do need to show you some of these adverse events. It was generally safe and well tolerated. The events were transient and simply related to dosing day. Vast majority of them were mild or moderate. There was a dose-related increase in the treatment emergent adverse events. So again, suggesting that perhaps that 8 milligrams struck a nice balance between safety and efficacy. The blood pressure and heart rate increases that we observed came down after about an hour, and most people were ready for discharge about 90 minutes later. As you can see, the majority of the adverse events were in these top three categories of nausea, headache, and administrative site pain, which I would say nausea and headache are the two primary side effects of the vast majority of medications of this type. As I mentioned, there was the open label extension. So after people completed the initial phase, that was 3 different doses and blinded, everyone was given the opportunity to have a open label 12 milligram dose. It was meant to see if we could see additional decreases in the MADRS score with a second dose, and then again to look at the sustainability of that effect out to 8 weeks after that additional dose. Something not mentioned, but obviously important for any audience taking these results into consideration, are that both in the randomized phase and in the open label extension, participants are provided with psychological support by trained facilitators. And they're assessed at multiple times by blinded raters. The second dose did provide additional significant results, clinically meaningful and statistically significant. That effect was sustained out to 8 weeks. Again, looking at the performance of both the 8 milligram and the 12 milligram, we saw greater both responder and remitter rates in both of those two groups. But because of a higher frequency that was dose dependent of safety events in the 12 milligram arm, we did select the 8 milligram dose to move forward for our phase 3 program. And here are those treatment emergent adverse events that were similar to the randomized phase, mild and moderate in severity, transient in nature. Same kind of grouping of the top 3 kinds of side effects that we'd observed in that randomized phase. There was one serious drug-related AE that resolved with additional patient monitoring and support. And we saw with that higher dosage that the average time to meet readiness for discharge was about 2 hours. So now we're going to talk about the program I'm most familiar with. So I'll probably, have the most to say about it. This is VLS-01. It is a dimethyltryptamine oral buccal mucosal film or buccal mucosal film. It also, dimethyltryptamine acts as primarily a 5-HT2A receptor agonist. The reason we developed the buccal film is we felt that that was a really patient and provider-centered way of delivering the DMT, as opposed to something like IV or IM. And so when you think about what's the least invasive procedure a participant could have in a clinic, a Listerine type strip dissolving on the cheek is fairly low in terms of the burden on both the patient and the provider to perform. We had pretty positive phase 1B results, and I'll go over some of the early stage results in just a second, which led us to advance the product to Elumina, which is our current phase 2 program in TRD as well. So both BPL-003 and VLS-01 are in TRD at the moment. And we are still actively recruiting for this trial. Our target N is 142. So that's where I was saying at the beginning, there are opportunities to still participate. I'll show you some ways to get involved, both as we continue with VLS, as well as continuing as developing the BPL phase 3 program. We are hoping to be done with that study with top line results by the second half of this year. Once again, kind of the raison d'etre of, I always say that wrong, of AtaiBeckley at this point is finding those rapid acting and sustained effect products that can still fit within the clinically demonstrated structures that we've had so far with ketamine programs. And I see that there's something in the chat. Hopefully that's not directed to me, but if anyone wants to chime in and let me know that it is, I'd appreciate that. So going over those phase 1 results, what you see here is the design of our early safety study, which was there were 2 arms, and in both of those arms, the healthy volunteers got a single dose of DMT intravenously. It was a 30 milligram infusion over 57 minutes in treatment period 1. And then there was a washout, and then in the second period, the groups diverged only by the order that they received these various concentrations of the buccal film. So what you'll see is that the Cmax is achieved pretty quickly and in about 30 minutes. And it was proportional between the 30 milligram infusion compared to the 120 milligram and 160 milligram buccal film. So again, just like BPL, kind of honing in on two potential dosages. And then after about the peak at 30 to 45 minutes, we did see a taper of the effect at about 90 to 120 minutes. At the peak effect, 13 out of 14 participants in the 120 milligram cohort had a subjective intensity rating of greater than 7. So that was an indication to us that, again, it was matching the experience of the IV, but also not being overly intense for the participants to experience. So the takeaways here about the safety profile, once again, we had similar PK concentrations of the 120 and the 160 as compared to the 30 milligrams of the IV infusion. And we saw a slightly higher rate of safety events in the 160 milligram group as compared to the 120 milligram group, which is what led us to move the 120 milligram dose forward. The treatment emergent adverse events that were most common were, again, headache and nausea, that there was dissociation and euphoric mood. All were transient, most resolving on the day of dosing, and any of the blood pressure and heart rate increases we did see did resolve mostly within 90 minutes and without any additional intervention. Additionally, these phase 1 participants did report that they felt the product engendered reports of increased self-reflection, which is, again, why we felt confident about moving it forward to phase 2 in a treatment population. So that's where we are now, phase 2, the Elumina trial, double-blind, placebo-controlled, designed to assess the safety and efficacy of repeated doses of the buccal film in patients with TRD. So again, I told you there's going to be 142 patients. They're randomized 1-to-1 to a placebo or the 120 milligram buccal film strip. That goes to the end of our first treatment period. The primary outcome assessment is, again, around a month. We then have a blinded period of follow-up before everyone is re-randomized to an open label dose. And that open label dose is going to further tease out the differentiation between the 120 milligram dose versus the 60 milligram dose. So everyone in the study does get at least one dose of active drug. Some of the criteria here for inclusion, moderate to severe TRD. Again, patients must be willing to discontinue antidepressants. They can't have used psychedelics within 6 months of screening. We do provide psychological support by trained monitors, both pre and post dose. And again, because this study is ongoing, I don't have any data to show you. So moving on to our other product in development, EMP-01, is an oral tablet that is the R enantiomer of MDMA, and that target population is social anxiety disorder. So as I said, it's an oral formulation. We chose R-MDMA because we do believe, based on early animal and human models, that it has unique and dose-dependent subjective effects that make it a little bit different from racemic MDMA. I'll show you in a second some information on social anxiety disorder, but again, it's a disease of high unmet need. And I think one of the most important things to point out is that there have been no new molecules approved for social anxiety disorder in over 2 decades. Most of the medications that are used to treat it are just repurposed medications that were already on the market. So we haven't had any new treatments in over 20 years. We did a phase 1 study that demonstrated favorable safety and tolerability. And then in our 2A study recently completed, we're hoping to see those top line results this quarter, possibly next quarter, depending on how the analysis goes out. And again, the reason that we chose R-MDMA is we think that it has a lot of the same positive characteristics of racemic MDMA without some of the negative considerations of that S enantiomer, and I'll go into that a little bit more in a second. So just reviewing why this treatment is important. Anxiety is the #1 most common mental health disorder in the United States. Over 40 million people are suffering from it. People report a pretty high level of impairment in terms of how the disease affects them. And like I said, we've had no new molecules approved in over 20 years. The people that are getting treatment report a really low recovery rate. So about a third of people are going to achieve some remission of symptoms. back to that differentiation we were talking about. So again, a unique pharmacologic benefit compared to racemic MDMA and potentially lower risk for adverse effects. We can see here the two different enantiomers that are part of racemic MDMA. Compared to R-MDMA, it has less, or compared to S-MDMA and racemic, R-MDMA has a reduced amphetamine-like approach. It's only a partial agonist of the 5-HT2A receptors. You can see here that it has very similar profiling to racemic MDMA. S is down here. And then if we look at effects on the body, the central nervous system, we can see that R-MDMA has a much lower effect on body temperature and heart rate. So in our phase 1 study, it was generally well tolerated with no severe or serious adverse events. It was a single ascending dose study. Body temperature remained within a normal range across the cohort compared to some of the higher body temperatures that are observed with racemic MDMA. The pulse changes and blood pressure changes were within an expected range and only slightly dose dependent. There was no increase in suicidal ideation. And I think the notable side effects that stand out here is that one of the most commonly reported side effects of racemic MDMA is bruxism, or grinding of the teeth. And in that phase 1, only 1 of 24 participants experienced that side effect. We found that R-MDMA does have a kind of unique profile on certain elements of the emotional breakthrough inventory and self-compassion scales. So it does have an increased effect on a few of these items, and it's broken out here. This is taken on the day after dosing. So people are reporting statistically significant increases in emotional breakthroughs. Emotional breakthroughs have been shown to improve efficacy in depression and anxiety studies involving classical psychedelics. And we're really hoping that this is going to be part of the equation as we move the product forward. It has more of a profile like classical psychedelics in terms of on the altered states questionnaires. So this is looking at a few of those different dimensions. People had more similar experiences to classical psychedelics on an R-MDMA than they did on racemic in some of the qualities, including oceanic boundlessness, vigilance reduction, visionary reconstruction, dread of ego dissolution, and auditory alterations. So again, it's more psychedelic, still active at the 5-HT2A, but without some of the stimulating side effects that are observed with classic MDMA. All of that led us to the development of the 2A study that recently concluded. It was assessing the safety and efficacy of 225 milligram doses of EMP, which were, again, the dose that we arrived at after that single ascending dose finding study done in phase 1. All participants had to be diagnosed with social anxiety disorder. You can see here what the inclusion scale was. The primary endpoint was safety and tolerability out to day 43. We were also looking at subjective drug effect scales, change in other clinician-rated scales, and we're still examining the pharmacokinetics of the compound and its metabolites. That's all I really have to say about the drugs. I did want to say that there are a couple ways to get involved and learn more. So as I noted, BPL-003 has completed phase 2. We've had our end of phase 2 meeting. We're preparing right now for phase 3, finalizing the protocol. We're primarily engaged right now in site selection, and that's where I'm supporting the team. So I put my email here. If you are part of a clinic that wants to be considered as a site for BPL-003, I'd be happy to share the materials for you for that part of the process. If you want to learn more about the phase 2 program, I put the clinicaltrials.gov link here. For VLS-01, we are currently open in the US, Canada, Australia, and the UK. At this point, we're not seeking any additional sites, but there may be clinicians on the line here who are interested in hearing more about the study, or maybe interested in referring their patients to the study. So once again, I dropped the clinicaltrials.gov link there where you can learn more. For none of these studies do we have a Atai specific recruitment page. So all of the trial sites where you would reach out to refer your patients, and currently we have 2 active in Canada, would be listed on clinicaltrials.gov. And then finally, for EMP-01, like I said, that phase 2A recently wrapped up. We're aiming to have those top line results at the end of this quarter, maybe beginning of next quarter. So be on the lookout for that. But if you want to learn a little bit more about the study design itself, as well as the phase 1 program, you can go to this clinicaltrials.gov link. And that's all I have for you.
Wonderful. Thank you, Hailey. That was super interesting. Definitely like, more on the pharmaceutical side of things. Um, I have questions. Do other people have questions as well? You're brave. I know it was a lot, and it was mind melty. Um, so I'll start off with a question, and this is kind of because I'm less literate in the pharma kind of world. But there was one slide that I found really interesting because of that, and it was a slide where you were comparing with the MDMA, our MDMA product. Um, you were comparing it to other psychedelics, and I was curious about that. Was that something you? I think I saw psilocybin. It was there's a lot to look at, but I think you were comparing it to psilocybin and MDMA. And yeah, can you share more? Like, was that part of your study? Did you do that study, or just share more about that?
Yeah, that's a great question. And if we were, I could say that if we were in a post-approval environment, I would never be allowed to show you a comparison slide like that. But because we're still in the research phase, I can give you an idea of how to think about these molecules that are under development, right? So
Mhm.
we did not do a head-to-head, to my understanding to this date that has not been. I mean, Harriet DeWitt has done some like deception type protocols where people are given a variety of compounds and she assesses their effects. University of Chicago, if people don't know who Harriet DeWitt is, she's brilliant. You can go check out her publications. But she's the only person I know that really runs like multiple arms of a study to assess effects. So that slide is really a compound of the 5D-ASC being assessed in racemic MDMA studies, the 5D-ASC being assessed in psilocybin studies, and then being compared to what we saw with the RMDMA compound in our study. So it was not a separate study comparing all three of those. But the takeaway from that slide is that people had greater oceanic boundlessness. They had reduced fear of ego dissolution. They had more auditory alteration with some more of the classic kind of psychedelic effects as opposed to the stimulant effect more seen in racemic MDMA. So when you look at our MDMA, it tends to look more like psilocybin on those types of experiences as it does to racemic MDMA.
Okay, super interesting. Thank you. There's some questions in the chat. Kat asks, how diverse are the trials? Historically, it's been very white, very male dominated, which has naturally been limiting.
Yeah, that's a great question. And I don't have access to those data. I could absolutely, I mean, I'm sure that they've been published in at least the earlier phase papers, like the phase 1 paper for VLS would have that, as well as the early phase 1 and the first phase 2 paper for Beckley would have those. Since EMP, just wrapped up, we'll be seeing that, that table 1 result soon. Table 1's generally where you see all those demographics. The slides that I have don't include that, but I agree that's a really important. I've been in research for 16 years. That's always a really important question. How do we make the population of our studies look more like the population of our patients? It's always an ongoing effort in clinical research. And, I can say that we are in 35 cities across the US and Canada, 4 cities in the UK, and 2 cities in Australia. So hopefully we have the opportunity to capture a more diverse population. And looking at what's coming in for VLS, I think I am pleasantly surprised at how we are doing that.
Thank you. There's a good, hi Gordon. There's a question here, good question from Gordon. When you say they have proper guidance, psychedelic help, where do people get trained on DMT specific? Or is it the same principles across the board with psychedelic assisted psychotherapy?
Yeah, so there are a couple of vendors in the psychedelic space that have emerged as sort of the training programs. We have used Fluence, which does offer courses to the general public. I myself have taken a few courses through Fluence, not as a practitioner, but in psychopharmacology of psychedelics, for example. And so, generally what these programs do is there are background modules on the history of psychedelics, the research to date, the concepts of set and setting, which we know do affect safety and efficacy. And then, depending on what role people play in the study, the later modules are more tailored to that. So there is a requirement set forth by the FDA that there are two people in the room. The former requirement in the early Lycos studies was that the lead therapist was a doctoral level, and Lycos was able to set a standard for the industry where now the lead therapist is a master's level clinician so that it expands the possibility of who can sit in that role. And then facilitators are generally people with a behavioral health background, but they don't have to have a master's degree as long as they have in-clinic experience. So for the general public right now, all of all of the trial training programs are really sponsor specific. I would say Fluence, I mean, I don't work for Fluence and I don't get any like, compensation for saying this, but I think that they probably offer the greatest number and diversity of training programs for the general public. Um, you know, my opinion solely, not the opinion of AtaiBeckley, is that when these medicines get rolled out, the professional organizations that accredit the various kinds of practitioners who can be involved will likely have their own standards. And I think we've seen that with both of the APAs, AP Psychiatry and AP Psychology, are coming out with textbooks. There's increasing amounts of material at these conferences where CMEs are offered in psychedelic research and facilitation support. So that is definitely a fast moving space. And I think at the end of the day, what it's going to look like if any of these compounds get approved, we don't really know.
Uh,
Folks, you can
One
raise your
question
hand.
I think that goes along with Gordo's other question, the first question in the chat, where it was interesting to see at a medium dose and a high dose, you had a similar clinical effect of the drug, but I'm wondering if there was any difference in like the subjective experience or the psychedelic mystical experience or other subjective experience of how intense it was and whether or not that had any predictive power of results.
Yeah, so I think in both BP, so EMP, we did the first dose finding study. It was a phase 1 healthy volunteers. We arrived at the 225 milligram dose. Just that program is a little bit earlier in development. I think we'll get more data from the phase 2 as to how that played out in terms of efficacy in a treatment population. Like Research 101 for folks here, like phase 1 is generally done in healthy volunteers. So they're people who don't have an illness. And so something that may be challenging for someone who doesn't have a diagnosis, or something that may not be challenging for someone who doesn't have a diagnosis, may be more challenging for someone who does, and maybe vice versa. Also, background rate of psychedelic use, someone that's anticipating these changes to occur can also affect how they report how the compound affected them. So EMP is kind of still a developing story for both BPL and for VLS. We had these two doses. For BPL, it was the 8 milligram and 12 milligram. The efficacy looked the same, and it was the safety where we saw a difference. There was a greater increase of adverse effects in the 12 milligram arm, which is what led us to go with the 8 milligram. And it was basically the same story for 120 milligrams of the buccal film DMT in VLS versus 160 buccal film DMT. We chose, like when we were choosing I'm less familiar, let's say, with the dose finding that was done in the early phases of BPL, and I didn't include those phase 1 slides simply because this was meant to be like an update. So we have to go back to the phase 1 papers on those, on how they did the dose finding for BPL. But in VLS, both the 120 milligram dose and the 160 milligram dose mapped really well in a PK curve to the 30 milligram drip over an hour of intravenous DMT. And so we knew that we were getting about the same amount of drug into the body. at either of those doses as we got with the 30 milligram IV. And again, we saw fewer reports of adverse events in the lower arm, which is what led us to choose that. And so the subjective intensity rating is a scale of 10. And in the 160 milligram arm, we basically got 10 out of 10 in most of our participants. We did not think that a 10 out of 10 experience was going to be the most therapeutic when you roll it out to possibly people who are taking something like this for the first time. And so the dose that we went with consistently tested at a 7 out of 10 subjective intensity rating. So again, you had the blood concentrations that matched a really standard dose of high dose DMT and IV, and you saw fewer treatment emergent adverse effects. So that's how those decisions are made. I would say most people don't start at if I could draw an analog, like most people don't start at a hero's dose, right? And we sort of thought that maybe you don't need as much to have just as good of an effect. And maybe later we'll see something different once we conclude phase 2. Now that we're in a treatment population, I would say that it's likely the data could be different in a healthy volunteer versus treatment population. We don't know yet.
Hey, Haley, I just had one quick question.
Yeah.
Thanks for the presentation. I had a question about actually the phase 2 design for VLS and EMP, and was just curious as to why you guys chose to do 2 doses. I can't quite recall when the endpoint was, whether there was an endpoint in between the 2 doses or not. But I was just curious to know why that was decided that way.
Yeah. I think, so in BPL, we're still considering what the phase 3 is going to look like. But generally speaking, you see a separation from placebo at 1 dose, and you see a greater separation from placebo at 2 doses. And I would say that that's fairly consistent across. This was true when we looked at the Lykos data. This is true now looking at these 2 compounds. Like, I think, something happens where people are able to better land in the experience on the 2nd dose. Maybe it's less frightening. They know how to work with it. And so we do see this greater separation from placebo pretty much across the board. I would say that a multiple dose model, as opposed to a single dose model, is consistent with how ketamine is being approached. It's an induction model. You're getting a dramatic change in symptomatology relatively quickly. And then, will people go into maintenance phase? I mean, I think that's what all the sponsors are looking at right now. Would we redose at 3 months? Would we redose at 6 months? A lot of the sponsors right now have adaptive protocols around thresholds for giving additional doses. So I think it's really meant to aim for a clinical practice in which someone doesn't get better after 1. There's a moderate improvement, but could we get someone more better? Again, in BPL, after the open label extension, the rates of response and remission increased by like 30% in both of those categories. So we think 2 doses might do something a little bit more than just a single dose. And we want to make sure that there's flexibility for patients and providers to adapt to the patient's needs. Maybe they only need 1, but why wouldn't, anything we do in a trial could be used in a label. And so giving more doses enables the provider and the patient at the end of the day to have that greater flexibility, because that's how it was done in the trials.
Cool, thank you. And I was also actually wondering whether you had any suggestions for reading about R-MDMA. I just thought that the enantiomer stuff was fascinating, and I don't know much about it. So if you have anything, suggestions as to what to read, I would love some.
Yeah, if you want to, I think we're connected on LinkedIn. I can drop the citations from those slides into a message. Yeah.
Following up on the question. Oh,
yeah.
sorry. Who is speaking?
Oh, I was just going to say also, just off the top of my head, Bill Fantagrassi, he's at University of Arkansas, I think. He's done some work, particularly he's a preclinical guy, but he's done a lot of work at looking at various ratios of RNS because there are a number of companies in this space that have patented those various ratios in the way that like Adderall has patented ratios of the stimulants. And he's just one person I can think of off the top of my head. Matt Young, who's now the head of the MSL team at Compass, did a lot of work with MDMA in animals before he went to work for Compass. So those authors, you can probably find some good information on, like more than what will be in the slide, more than what I shared.
Yeah, it's an interesting point that Noah made. I mean, in terms of the two doses. So several clinical trials, a lot of them will have two doses, and that does give more flexibility in the clinical space. But where it doesn't is they can't get three doses because there are no clinical trials. So I'm just curious, just more generally, not, for anybody, but just, do any clinical trials do more than two doses?
Yeah,
Are there
yeah, no,
implications for more than two doses?
yeah, well, both of the psilocybin sponsors have open label extensions in their current phase 3 protocols, which enable redosing if there's not been remission or if there's a relapse. I believe MindMed also, I mean, MindMed is single dose. So I think that they're adding a multiple dose open label extension onto their phase 3s, which is part of what's taking them so long, is these OLAs generally extend out over about a year. Whereas, when you're looking at the 1 or 2 dose induction model, your primary outcome is maybe at day 29. We follow up to 8 weeks. The FDA's made it really clear they want to see durability. I think that was a learning from Lykos, right? Lykos didn't have this redose protocol. And so I would say almost every other sponsor that I'm aware of is looking at how often, and they've written a protocol that gives the investigator the ability to decide with the patient, do you want another dose? Do we think another dose is needed? Obviously one of the endpoints too is capturing who goes back on rescue medication. If they didn't respond to the antidepressants and that's how they got into the trial, do they have a different level of response now that they've been given this intervention? So I think we're going there.
Yeah. And I'm going to throw in another one. I'm just following up on Gordon's, his one. So, with the training I wasn't clear that the people like had to have training specifically on psychedelics or DMT. But also I'm thinking about the aftercare, for people who, yeah, just what the aftercare looks like, and just thinking about what you just said, people who were treatment resistant, and if this doesn't work, there can be a real, like suffering and loss of hope. So I'm just wondering what kind of care comes for the people after the trial.
Yeah, I mean, in every trial that I've worked on, there is, a point at study exit. I mean, in every trial I've worked on, there is a baseline number of uh, prep and integration sessions that occur as part of the protocol. At any time, a participant who needs more support can request that, and the trial will honor that, right? And that was, again, mentioned in the Lykos data, how many people got more support than what was written into the protocol in the FDA's critique. After the trial ends, I think that it's sort of an interesting real world case. I mean, the nature of these trials is that they do end. They have to have an end point because there's limited funding to carry, that kind of protocol on. We have to answer a question that fits within, what are these sort of industry regulated time points? And generally, like the FDA says, how do you look at 30 days? After a month, what does the treatment look like in terms of efficacy? I think there's a lot of really incredible participant-led advocacy for this ongoing support. Pedram Dara has done a whole conference now on the lived experience of participants who are in these trials, and he's organized groups where people can come together. Pedram's Canadian. You guys probably know him. So, I think that there's often a way in which science is limited that community shows up. And in Europe, Saipan is doing a similar thing. I think if I have to think about, again, what this looks like potentially in a post-approval environment is, I mean, I would hope that I give the example of, I worked on PrEP, pre-exposure prophylaxis, and that really started as a very clinician-based intervention in our clinical trials. It is now very much a peer-supported intervention. So there are peer healthcare navigators who help people figure out how to get it paid for. They talk about the challenges of the adherence. And so, I see that happening with the research participants, and I wonder if that sort of thing will happen in the real world if these molecules get approved. I think that there are a lot of examples of bundled care getting approved. I mean, this is where I feel a little bit ignorant about, Canadian healthcare structures, but there's a lot of groups right now really interested in pursuing bundled care approaches with ketamine, and people have been very successful in getting that covered by CMS, which is the US Medicare and Medicaid system in 6 states so far. So you get an infusion and you get the concomitant therapy. So I think that structures will emerge, and we just don't know what they are yet. But I believe that they will come. We got to get one out there first, and then we have our laboratory.
I have two like kind of random questions. One, how big roughly is like the DMT trial of psychedelics? Like if it's like 80% is psilocybin and 10% MDMA or whatever the metrics are, how much stuff is getting researched and put out? How much of that do you think is like DMT?
Oh, we're definitely a really, small representation. I would refer you to, if you don't already follow him, he's my good friend and genius, Josh Hardman from Psychedelic Alpha. He has a bullseye that discusses like what companies are in which stage researching which molecules. I've never actually counted. I can think of like Cybin had an IM DMT compound. Obviously GH is BPL's biggest competitor. They have an inhaled version of 5-MeO-DMT. There are people who are looking at smoked DMT in Brazil, but I don't think that's being done by a pharmaceutical company. I think that's that's like a researcher-led trial. But definitely this is among the minority of the compounds there. I think there are far more in psilocybin, and no one's really working with LSD other than MindMed. I don't know, but
Thank you. And the other question would be kind of like what Riel was talking about, the pre and post. I know that has nothing to do with kind of what we're talking about right now, but is there not any uh wouldn't or shouldn't that be a standardized thing if you guys are taking like metrics? Like what if one preparation is actually better than someone else's preparation, and same with on the other side? Couldn't that alter it? And is there a standard that everyone goes by?
you mean between compounds or like within the study?
Yeah, between compounds. Like if everyone's testing psychedelics, but everyone's just doing whatever they want for preparation and integration, it's like, aren't you gonna have a variant in that in itself? And couldn't there be like a standard, or should there be a standard?
Yeah, I think that I would say that all psychedelic sponsors have some version that builds upon the inherited principles from the underground and early trials of there is preparation with psychoeducation on the effects, there is intention setting, and there is support during the experience, which is meant to be non-directive. It's meant to be sort of, positive regard, affirmation of the experience and for the participant safety. And then in integration, it's supported meaning making, right? But it really shouldn't be directive. And I can at least speak to, I'd say, 3 of the major sponsors do analysis of the recordings to assess fidelity to the therapeutic model that they've trained and kind of certified their facilitators in. So of the major sponsors, I'd say most of them are assessing, did they do this, this, and this to support what we think is should be the standard approach. Obviously practices vary widely in the underground. I think, as I mentioned, the agencies that approve these applications really only reviewed drugs. Like we kind of broke the mold, or Lycos broke the mold with trying to propose this therapy component as being part of it, because it's just not something that the agency really is accustomed to reviewing how much that contributes to it. I mean, there are other drugs that were approved with behavioral interventions, like Suboxone. PrEP, when it was first rolled out, had to have behavioral counseling around risk reduction. People were afraid that there were going to be a lot of breakthrough HIV infections because people were going to change their risk patterns. That being said, again, like we wanted to leave it as, we are leaving it as broad as possible about who can be the facilitators in the room. All of those people have to be licensed. That will be a requirement of any REMS program that the FDA allows for these products, and that's risk evaluation and mitigation strategy. So everyone will have to be licensed. People are licensed by bodies such as the American Medical Association for Doctors, or like American Psychiatric Association. You can be board certified through various specialties. And the hope is that if you're a MFT, who you got your licensure for, will establish those standards for what an MFT does based on what we've demonstrated in clinical trials as being like the essential elements. If you're an MD, those will be part of your, if you want to deliver this kind of medicine, right, that those will be the standards of care that you'll be required to adhere to. I think the challenge in psychedelics is that they're all very different. All the molecules are very different. I think there are very core elements of the kind of support that is provided in all of these trials, which I've already named, but some of them have more, again, psychedelic effects, some of them more emotional effects. They're different durations. And so I think to say that like there's one approach really that like fits all of the molecules, I think we can really only go by what people's discipline and training is in. What level of care are people actually licensed to provide? And that's based on the kind of credentials that they have. And so that's where I think that those governing bodies will have to determine, okay, you're an RN that does this versus you're a massage therapist, if we got there, right, that does this, or you're an MD that does this. And they operate within their scope of practice. And an MD or an RN would not provide therapy. They're not trained to do that.
I was trying to find the button to raise my hand. I don't know how. I have one more question. You had kind of touched on community and how community can kind of support the efforts of science and then the things that are happening in science. And the MAPS Canada community is extensive and enthusiastic. So we have our folks here today, and then this webinar gets recorded and goes out into the world, and then more people will watch it. And so I'm curious, just what are some of the biggest challenges right now that you're facing in the clinical trials phase and coming up to the commercialization phase? And can you think of anything that we could do to help?
Well, Canada's already leading the way. You guys have special access program. Like, so you guys are collecting real world data and rolling it out in a meaningful way within the structure of your own healthcare system that, not many other countries have the opportunity to do. Switzerland, Australia, that's it, right? It's like those three. So I think that there's going to be a lot of opportunity to collect data in those programs that can inform, I think, when the big sponsors go to launch, what worked, what didn't, what was cost effective, what types of care that were provided supported the best outcomes because research is limited, because research has limited dollars. I think that one of the biggest challenges that the entire industry faces is hype and expectation, right? And so we've gotten better as an industry about measuring expectancy at baseline. And so I think that the extent to which people who are advocates in this space and researchers in this space are careful with their language I think that's also, I wanted to raise that, Rio, when you asked the question about 1 dose versus 2. I think it would be misleading. It would be an inaccurate representation of how these medicines have been used to call them like one and dones and cures. I think we can get to really strong levels of functional recovery. But that's why the 2-dose induction model, I think, is important, and looking at maintenance dosing is important. Because otherwise we've led people to believe that something might happen that we've never actually really seen happen in any of the trials, right? Miraculous sort of recoveries. So being careful in our language around hype and expectancy. There are no silver bullets. Nothing is 100% effective. We're just hoping to create a margin of efficacy over the additional of the treatments that are currently available that has, arguably less or the same safety concerns. We need sites in Canada for sure. So anyone who wants to engage with the BPL site selection process, I do think that you guys have a great industry and again, like you said, a passionate and dedicated group of researchers. And there really is little representation. We had 4 sites in Canada compared to a much larger number in the US. And so getting more Canadian sites involved, I think, is definitely on our agenda for the next phases. We don't have an investigator-sponsored research program at AtaiBeckley yet. I think that's a question that comes up. But the psilocybin companies, the MDMA companies, maybe the LSD companies are all doing investigator-sponsored research. So if people are seeking product, those are opportunities. We don't have those yet. So they are on the website, the clinicaltrials.gov website, where those sites are in Canada.
And we can also put them on our website.
Yeah, I can, the slides that I shared are generally available on our website. I adapted them for this audience, but the one that I actually created was like the list of links. So I'd be happy to share that one slide over email, because that's the only differentiator really from the rest of the presentation. And that way everyone has it.
Sonia, do you know if we put that on our website yet? I know we put it in our newsletter for the recruitment, but
We are working on it. I saw that a list went up today and we're reviewing it, so we're working on it.
Okay, so that will be on our, we're trying to put sites that are recruiting in Canada on our website for ease. We're working on a database that will have that, but right now we're just listing them. So thank you. Are there any final questions? Okay, I'm going to share my slides again and have a big thank you. I want to share some links if I can. I don't know if I can in the chat. Here we go. And some links that you can contact us. I want to thank Hailey and everyone for coming today. Our next session is on February 16th with Dr. Ann Wagner, who's doing couples therapy with MDMA, conjoint couples therapy. With MDMA, that's right. And I invite you to join our mailing list to get up to date information about registration for all our initiatives that are coming up. You can visit our webpage for today's recording. We'll link it to a couple places where you can find it, as well as our past research room presentations. Email us if you want to present or want to see somebody else present. And we really appreciate your attendance today. Thank you so much, Hailey and everyone.
Thank you.
Thank you very much.